Welcome, curious minds, to another deep dive. Today we're embarking on really one of the most fundamental journeys in all of biology and medicine. We're looking at how our bodies take the genetic blueprint, the ultimate instruction manual, and actually use it to build, well, everything. Every single protein, every cell, basically every part of who we are. We're exploring gene expression and the synthesis of proteins. It's a deep dive into this incredible dance between DNA, RNA, and all the intricate cellular machinery that brings life's blueprint to fruition. And it's just a cornerstone of modern medicine, really, driving everything from, new drug discoveries to our deepest understanding of disease.
Exactly. This area isn't just like theoretical stuff for textbooks. It's the absolute bedrock of molecular biology. And grasping these mechanisms gives you essential insights into how countless drugs actually achieve their effects, and also why even a tiny, tiny alteration in our genetic code can lead to pretty profound illness. So our goal today is to meticulously trace that path, right? Starting from the genetic material itself, DNA, and going all the way to the functional proteins that are essentially the workhorses carrying out almost every task in every cell.
Okay, so what's our mission for this deep dive then? We're going to guide you through the major concepts, the really fascinating biochemical pathways, and maybe touch on some compelling clinical examples too. Think of this as your essential shortcut maybe to understanding the intricate flow of information, from genes to proteins, seeing how each piece fits together to form this cohesive, absolutely vital picture, ready to unravel the secrets of life's instruction manual. Let's dive in. All right, so to truly appreciate how our genes get expressed, how this whole thing works, we have to start right at the very foundation. We need to understand the molecules that actually carry this genetic information. So we're talking about Chapter 12 here, structure of the nucleic acids.
Indeed, we have to start there. We're talking about DNA and RNA, and their fundamental building blocks, the monomers, are called nucleotides. You can sort of imagine a nucleotide as a three-part molecular Lego brick, if you like. Each one has a nitrogenous base. These are the letters, the A, G, C, and T in DNA, or A, G, C, and U in RNA. The purines are adenine A and guanine G, and the pyrimidines are cytosine C, thymine T, or uracil U. Then there's a 5-carbon sugar. And finally, you need at least one phosphate group attached. These seemingly simple units, they're the basic currency of all genetic information.
So, okay, they're both made of these nucleotide bricks. But what are the critical distinctions? What really sets DNA and RNA apart at this basic building block level?
Yeah, the key differences, they might seem subtle, but they have really profound implications for how these molecules function. They mostly lie in the sugar component and in one of the pyrimidine bases. So in DNA, the sugar is deoxyribose. The deoxy part literally means it's a ribose sugar that's missing a hydroxyl group, an OH group, on its 2' carbon atom.
Deoxy, missing oxygen.
Right. RNA, on the other hand, it keeps that hydroxyl group, so it contains just plain ribose sugar. And here's the other distinct feature. Where DNA uses the base thymine T, RNA substitutes uracil U in its place.
Right, U instead of T in RNA.
Exactly. So DNA's alphabet is A, G, C, and tRNA's is A, G, C, and U. And these, seemingly minor structural differences profoundly influence their stability, how they fold, and their very distinct roles in the cell.
And then there's the grand architecture, especially for DNA. This is where it gets really iconic, doesn't it?
Yeah.
How is DNA typically structured to not only store but also protect all this vital information so effectively?
DNA, yeah, the image most of us have is spot on. It's famously a double-stranded molecule spiraling into that beautiful structure we call a double helix. Picture 2 incredibly long, intertwined polynucleotide strands. They're coiled around a central axis, much like a meticulously crafted spiral staircase.
Okay.
Now, these 2 strands aren't just floating next to each other. They're intricately joined by relatively weak hydrogen bonds that form directly between their nitrogenous bases. And this bonding isn't random at all. It follows very strict rules of bis pairing. Adenine A always pairs up with thymine T. They form 2 hydrogen bonds. And guanine always pairs with cytosine C. They form 3 hydrogen bonds.
A with T, G with C. Always.
Always. This complementary pairing, it's absolutely fundamental. It's the key to how genetic information is accurately copied and then transmitted across generations. Meanwhile, the sugar and phosphate groups, they link together covalently to form the sturdy, repeating sugar-phosphate backbone of each strand. This provides the structural integrity, the frame, for the entire helix.
That spiral staircase analogy really clicks for me. So the sugar-phosphate backbone forms the elegant railings, and those perfectly paired bases are the steps connecting the 2 sides. Is that a fair way to imagine it?
That's a perfect analogy, actually. And a crucial detail that often gets glossed over is that these two strands run in anti-parallel directions.
Anti-parallel.
Yeah, imagine one strand has a chemical directionality. We call it 5' to 3', 5 to
Okay.
3. Its partner strand runs in the exact opposite orientation, 3' to 5', 3 to 5'. They run head to tail, essentially. And beyond those hydrogen bonds holding the pairs together, the flat stacked base pairs themselves interact. They engage in subtle electron interactions, generating what we call hydrophobic stacking forces. These stacking forces, along with the hydrogen bonds, are the primary contributors to the remarkable stability of this double helix structure. It's surprisingly stable.
Hmm.
And if you were to look closely at the surface of this helix, you'd also notice these alternating indentations or grooves. We call them the major groove and the minor groove. These grooves aren't just, surface features. They are vital points of interaction. They act as recognition sites where other molecules, particularly proteins involved in gene regulation, can bind and read specific DNA sequences without having to unzip the entire helix. It's quite clever. And while the most common form in our cells is the right-handed bDNA, it's worth noting there are other forms too, like aDNA and zDNA. They differ in things like their width and even their handedness. zDNA is left-handed. It just shows the structural versatility DNA can adopt.
Fascinating. So we have this incredibly stable, elegant, double-stranded DNA structure. But what about RNA? How does its structure compare to this elaborate double helix, and how does that difference influence its roles in the cell?
Right, so unlike DNA, RNA is typically single-stranded. But, and this is important, don't let single-stranded fool you into thinking it's just a simple floppy molecule. Not at all. RNA molecules are incredibly dynamic. They can fold back on themselves and form really intricate, complex, three-dimensional structures. And they do this through intramolecular base pairing between bases on the same strand.
So it pairs with itself.
Exactly. This internal pairing creates fascinating structural motifs, things like loops and stems and hairpins. And these structures are absolutely crucial for RNA's incredibly diverse functions. It allows RNA to act sometimes as an enzyme, or as a structural scaffold, or of course as a messenger. Now, within the context of gene expression, the process we're focusing on today, we mainly talk about three major types of RNA. Each one plays a very distinct role in protein synthesis.
Okay, what are the big three?
First, there's messenger RNA, or mRNA. This molecule acts as the vital intermediary, the go-between. It carries the specific genetic information, the actual recipe for one specific protein from the DNA blueprint, which is safely stored in the nucleus, out to the cytoplasm where the protein factories are.
So it's the messenger carrying the order.
Precisely. It serves as the precise template for building proteins. And in eukaryotes, like us, mRNA isn't just a raw copy. It undergoes significant processing before it leaves the nucleus. It gets a distinctive 5' cap added to one end and a long string of adenine nucleotides called a poly-A tail added to the other end.
A cap and a tail.
Yeah, a cap and a tail. These modifications are really important. They protect the mRNA from being chewed up by enzymes, they help it get transported out of the nucleus, and they're crucial signals for the machinery that will eventually translate it into protein. And the core message within the mRNA is contained in its coding region. This region is read in sequential sets of 3 bases, known as codons. Each codon specifies a particular amino acid.
Okay, mRNA carries the code. What's next?
Second, we have ribosomal RNA, or rRNA. Now, rRNA isn't just a messenger. It's actually a major structural and catalytic component of the ribosomes.
The protein factories themselves.
Exactly. Think of ribosomes as the cellular machines where protein synthesis actually happens. rRNA molecules combine with many ribosomal proteins to form these factories. And these factories differ slightly between different forms of life. Prokaryotic ribosomes are a bit smaller. We classify them as 70S, and they're made of a 50S and a 30S subunit. Eukaryotic ribosomes in our cytoplasm are larger 80S with 60S and 40S subunits. Interestingly, the ribosomes inside our own mitochondria, the cell's powerhouses, actually look more like prokaryotic ribosomes. That's a really powerful piece of evolutionary evidence, by the way.
Huh, fascinating echo. Okay, mRNA, rRNA. What's the third one?
And third, there's transfer RNA, or tRNA. These are the crucial adapters, the interpreters in protein synthesis. Each tRNA molecule is like a specialized delivery truck. It's designed to carry one specific type of amino acid, the building blocks of proteins, to the ribosome factory.
A delivery truck for amino acids. I like that.
Yeah. And it has a very distinctive structure, often drawn in 2D as a cloverleaf structure. More importantly, it has a unique 3-base sequence on one of its loops called the anticodon. This anticodon is designed to precisely bind, through base pairing, to a complementary codon sequence on the mRNA molecule that's being read by the ribosome. This tRNA-mRNA interaction is what ensures that the correct amino acid is added to the growing protein chain in the exact order specified by the mRNA blueprint. It's like matching a key, the anticodon, to a specific lock, the codon. Incredible precision.
That's a tremendous amount of intricate structure packed into these nucleic acid molecules. It really is amazing. But it makes me wonder, okay, why should we, from a medical perspective, care so deeply about all this detailed molecular architecture? How does this deep dive into nucleic acid structure become immediately relevant to, say, treating patients?
That's the crucial link, isn't it? It's one thing to marvel at these structures in a textbook, but the real aha moment for us in medicine comes when we see how this fundamental knowledge directly impacts diagnosis and, importantly, treatment. Consider the drug zidovudine, better known as AZT. It was one of the first really effective treatments for HIV.
Right, AZT for HIV.
AZT is a fantastic example of a nucleoside analog. It's synthetically designed to mimic a natural thymine nucleotide, one of the building blocks of DNA. Now, HIV is a retrovirus, meaning it uses an enzyme called reverse transcriptase to copy its RNA genome into DNA. When AZT gets incorporated into this growing viral DNA strand by the reverse transcriptase, it abruptly stops the chain from getting any longer.
It just stops the cold. Right.
Because AZT is cleverly designed to lack the necessary 3' hydroxyl group, that OH group we mentioned earlier on the sugar, DNA polymerase, or in this case, reverse transcriptase, absolutely needs that 3-OH group to form the next chemical bond, the phosphodiester bond, to add the next nucleotide. AZT doesn't have it, so it acts as a chain terminator. It's a chemical dead end specifically for the viral replication process. This is just a brilliant example of how understanding nucleic acid structure at such a granular level allows us to design drugs that specifically interfere with viral replication, saving countless lives.
That really is a powerful connection, a clear example of intelligent drug design based on fundamental biochemistry. And we also encountered how certain chemotherapy drugs work this way too, right? Like doxorubicin, which is often used in cancer treatment.
Exactly. Doxorubicin exerts its potent anti-cancer effects through this very understanding. It works by a process called intercalation.
Intercalation.
Yeah, it literally means it wedges itself, slides in between the stacked base pairs of the DNA double helix, like putting a book between shelves that are too close together. This intercalation physically distorts the normal, stable structure of the DNA helix. By doing so, it effectively jams up the molecular machinery that needs to interact with the DNA, particularly the enzymes responsible for both DNA replication and the transcription of genes into RNA.
So it blocks both copying the DNA and reading the genes.
Precisely. This disruption ultimately leads to the death of rapidly dividing cells, which is characteristic of many cancer cells. So it really hammers home how this foundational understanding of DNA structure isn't just academic, it's absolutely key to developing effective treatments for a whole spectrum of devastating diseases, from viral infections like HIV to aggressive cancers.
All right, so we've established the intricate blueprint DNA, and we understand its elaborate structure. But, a blueprint is kind of useless if it can't be reproduced perfectly, reliably, time and time again, especially when cells divide. So how do cells manage to make exact, faithful copies of this incredible genetic instruction manual? This brings us, I think, to Chapter 13 and the astonishing process of DNA replication.
Replication is indeed the process where the two parental DNA strands serve as meticulous templates for the synthesis of new complementary DNA copies. It's an incredibly precise and really fundamental process that has to happen every single time a cell divides. And what's truly elegant about it, biologically speaking, is that it's famously semi-conservative.
Semi-conservative. Okay, you mentioned that before. What does that mean exactly?
It means that each new DNA molecule produced isn't entirely new from scratch. Instead, it consists of one original parental strand conserved from the original molecule and one newly synthesized strand built upon that template. It's a remarkable way of ensuring fidelity. You're keeping half of the old, proven information, hence semi-conservative, which inherently reduces the chance of errors creeping in during the copying process.
That's a brilliant strategy for ensuring accuracy. Okay, let's try to unpack this complex process. Maybe we can start with simpler organisms again. What's happening in prokaryotes, like a bacterium, say E. coli, when it needs to copy its DNA?
Right. Prokaryotes provide a slightly simpler model to start with. In bacteria like E. coli, replication kicks off at a single specific site on their typically circular chromosome. This site is called the origin of replication, often abbreviated as OREC. From this single starting point, replication proceeds bidirectionally. That means two replication forks. These are the points where the DNA is actively being unwound and copied, move out in opposite directions around the circular chromosome, unwinding and synthesizing DNA as they go, like two dippers opening up the circle from one point.
Okay, two forks moving outwards.
Exactly. And it's a highly coordinated effort involving a whole ensemble, a team of specialized proteins. First, you have enzymes called helicases. These are like molecular zippers. They move along the DNA using energy to unwind the parental double helix and separate the two strands.
Unzipping the DNA. Got it.
But as you unzip, imagine twisting a rope. It creates tension ahead of the unwinding point. This torsional stress or supercoiling could potentially stall replication. So you need enzymes called topoisomerases. A well-known one in bacteria is DNA gyrase. Topoisomerases act like molecular stress relievers. They temporarily cut one or both DNA strands, allow them to untwist, and then rejoin them, alleviating that supercoiling stress. Very clever.
Like letting the rope untwist.
Precisely. Then, as soon as the strands are separated by helicase, single-strand binding proteins, SSBs, quickly coat the exposed single strands. Their job is crucial. They prevent the separated strands from snapping back together, from reannealing, which would obviously block the polymerase from doing their job. They keep the strands available.
Keep them apart.
Next up is an enzyme called primase. This is important because DNA polymerase, the main builder enzyme, has a limitation. It cannot initiate a new DNA strand from scratch. It needs a starting point, specifically a free 3' hydroxyl group. Primase solves this by synthesizing short stretches of RNA primers, maybe 10, 12 nucleotides long, directly onto the template DNA. These RNA primers provide that necessary 308 starting point for DNA polymerase to latch onto and begin synthesizing DNA.
So it needs a little RNA starter piece.
Exactly. And now we get to the main architects, the DNA polymerases. E. coli has three main ones, creatively named POL3, POL2, and POL3. DNA polymerase of the third, POL3, is the primary replicative enzyme. It's the real workhorse, synthesizing the vast majority of the new DNA. And it always works by adding nucleotides to the 3' end, so it synthesizes in the 5 to 3' direction. DNA polymerase POL9 has a more specialized role, kind of like cleanup and patching. It comes in later to remove those RNA primers laid down by primase and then fills in the resulting gaps with DNA nucleotides. Importantly, POLIA also has proofreading activity, which helps fix errors. POL2 is mostly involved in DNA repair. And finally, after all the pieces are synthesized and the gaps are filled, you have little nicks or breaks left in the sugar-phosphate backbone between adjacent fragments. DNA ligase acts as the molecular glue, sealing these nicks by forming the final phosphodiester bonds, joining all the newly synthesized fragments together into continuous seamless strands.
Okay, that's quite the molecular machine. So if I'm picturing this replication fork, this Y-shaped structure where the DNA is splitting, you mentioned pol third only synthesizes 5 to 3 minute, but the two template strands are anti-parallel. How does it work? You must have one strand, the leading strand, where synthesis can just follow the fork continuously. But the other strand, the lagging strand, must be synthesized differently, right? Maybe in short pieces. Those are the Okazaki fragments.
Precisely. You've hit on a key aspect of replication. Because the two template strands are anti-parallel and DNA POL3 can only synthesize in the 5 to 3 direction, the two new strands are made differently. On the leading strand, the template runs 3 to 5 minutes. This allows pol third to synthesize the new strand continuously in its preferred 5 to 3 minute direction, simply chasing the replication fork as it opens up the DNA. One long, continuous piece.
Smooth sailing on that side.
Right. But on the lagging strand, the template runs 5 to 3. Since Paul III must synthesize 5 to 3 mRNA, it has to work kind of backward, discontinuously, moving away from the direction the replication fork is opening. It synthesizes short segments, maybe 1,000, 2,000 bases long in bacteria, called Okazaki fragments. Each of these fragments starts with its own RNA primer laid down by primase.
So it's like backstitching, almost little segments made backwards.
That's a good way to think about it. Then, after these fragments are made, POL-I comes in. It removes the RNA primer from the beginning of each Okazaki fragment and replaces it with DNA. And finally, DNA ligase seals the nick between the end of one Okazaki fragment and the beginning of the next one, joining them all together into a complete continuous lagging strand. It's definitely more intricate, but it's an incredibly efficient solution to the anti-parallel problem.
Wow.
And it's really worth emphasizing again that the main DNA polymerases, like pol III and pol I, also possess remarkable proofreading capabilities. Specifically, they have a 3 to 5 exonuclease activity.
Exonuclease, like cutting from the end.
Exactly. It means if the polymerase accidentally adds an incorrect nucleotide, one that doesn't base pair properly, it can sense the mistake, back up one step, cut out the wrong nucleotide using this 3 to 5 fit cutting activity, and then try again with the correct one. It's like a built-in delete key or undo button. This proofreading function significantly reduces the error rate during replication, making it incredibly accurate, maybe only one mistake per billion base pairs copied, which is astonishing, but still not absolutely perfect.
Yeah, astonishing accuracy. But still, not zero errors. And what's truly amazing, or maybe scary, is that even with all that sophisticated proofreading, mistakes can still slip through. And those uncorrected errors, those changes in the DNA sequence, are what we call mutations.
Indeed. Errors happen. And mutations are the raw material for evolution, but they can also be the cause of genetic diseases. And this whole complex picture we've painted for bacteria, it raises an important question, doesn't it? How do eukaryotes organisms like us, with our vastly larger genomes, our DNA packaged into multiple linear chromosomes inside a nucleus? How do we manage this incredibly intricate and absolutely vital process of DNA synthesis? It's a significantly bigger challenge in terms of scale and organization.
That's a really good point. Yeah. How do we handle it? What does eukaryotic DNA synthesis look like compared to the prokaryotic system we just discussed? What are the key adaptations for our complexity?
Right. So in eukaryotes, DNA synthesis is exquisitely timed and tightly linked to the cell cycle. It doesn't just happen whenever. It primarily occurs during a specific window called the S phase, or synthesis phase, which is nestled between growth phases G1 and G2. During S phase, the cell meticulously duplicates its entire genome in preparation for eventual cell division, mitosis.
So it's carefully scheduled.
Very carefully scheduled. And unlike prokaryotes with their single origin of replication on a circular chromosome, eukaryotic chromosomes are much larger and linear. To replicate these massive amounts of DNA within the time frame of S phase, eukaryotes utilize multiple origins of replication scattered along the length of each chromosome. This strategy initiates replication simultaneously at many points, creating numerous replication bubbles that expand and eventually merge. This massively speeds up the overall process of replicating the entire genome.
Okay, multiple starting points make sense for bigger genomes. What about the enzymes? Do we have the same polymerases as bacteria?
No, eukaryotes employ a far more diverse and specialized arsenal of DNA polymerases. It's not just pol first, 2, and 3. Think of it more like a finely tuned orchestra, with different polymerases playing highly specific roles. The nomenclature gets a bit complex with Greek letters, but the main players are polymerase alpha, polo. This one acts as the initiator. It works together with primus. Primus lays down the RNA primer, and then Pol alpha extends it with a short stretch of DNA nucleotides, basically getting things started on both the leading and lagging strands.
The starter polymerase.
Kinda, yeah. Then it hands off the job. Polymerase delta, polo. This is generally considered a major polymerase, primarily responsible for synthesizing the lagging strand, extending those Okazaki fragments after Pol alpha has started them. It's highly processive, meaning it can add many nucleotides without falling off. Polymerase epsilon, polo. This is thought to be the major polymerase for synthesizing the leading strand continuously. Like pol delta, it's also very processive and has proofreading capability.
Okay, delta for lagging, epsilon for leading, mostly.
Generally, yeah, though there's still some debate on the exact division of labor. Then there's polymerase gamma, pol-A. This one is quite unique. It's located exclusively in the mitochondria and is responsible for replicating the mitochondrial DNA, that small circular genome we have in our cellular powerhouses.
Right. Mitochondria have their own DNA.
They do. And beyond these main replicative polymerases, there's a whole collection of other specialized polymerases. We have pol beta, pol eta, pol iota, pol zeta, pol kappa, pol lambda. These are mostly involved in various DNA repair processes. They act like specialized mechanics, fixing different kinds of DNA damage. Some are called translesion or bypass polymerases because they have the unusual ability to synthesize DNA across a damaged template that would normally block the main polymerases, albeit sometimes with lower accuracy.
Wow, a whole toolkit of polymerases.
It really is. And another crucial eukaryotic protein, not a polymerase itself, but essential for replication efficiency, is pCNA. That stands for proliferating cell nuclear antigen. pCNA forms a ring-like structure that acts as a sliding clamp. It literally encircles the DNA strand and tethers the main replicative polymerases, like pol delta and epsilon, to the template. This prevents the polymerase from dissociating, making it much more processive, allowing it to synthesize much longer stretches of DNA without falling off. Think of it like keeping the train firmly on the tracks.
A sliding clamp keeping the polymerase attached. Makes sense. And then you mentioned linear chromosomes. That creates a special problem at the ends, doesn't it? Bacteria with the circular DNA don't have ends, but we do. The telomeres.
Yes, the telomeres. They are indeed a fascinating eukaryotic adaptation and a critical area of research, particularly in aging and cancer. Telomeres are specialized structures found at the very ends of our linear chromosomes. They consist of long stretches of repetitive DNA sequences. In humans, it's TTJG repeated thousands of times, along with associated proteins. Their crucial function is to act like protective caps, like the plastic tips on shoelaces.
Protective caps? How?
They protect the vital genetic information within the chromosome from being progressively lost during each round of replication. The problem arises because of how the lagging strand is synthesized. When the RNA primer at the very 5' end of the lagging strand is removed, there's no way for conventional DNA polymerase to fill that gap because there's no upstream 3'OH group to extend from.
Ah, the end replication problem.
Exactly. This means that with each round of replication, the lagging strand would get slightly shorter, progressively eroding the chromosome ends. Telomeres act as a buffer zone of non-coding repetitive DNA so that the shortening eats into the telomere repeats rather than essential genes.
But can that go on forever? Don't the telomeres eventually run out?
In most of our normal somatic cells, yes, they do. The enzyme responsible for counteracting the shortening, called telomerase, is typically not very active. Telomerase is a really unique enzyme. It's a reverse transcriptase, specifically an RNA-dependent DNA polymerase. It actually carries its own RNA template within its structure. It uses this RNA template to synthesize additional repeats of the telomeric DNA sequence onto the chromosome ends, effectively extending them and counteracting the shortening that occurs during replication.
So telomerase can rebuild the ends.
It can. But here's the crucial part for health and disease.
Yeah, this is where it gets really pivotal, doesn't it? Telomerase activity is usually very low or even completely absent in most of our differentiated normal somatic cells. This means our telomeres do gradually shorten with each cell division. They act as a kind of cellular clock, counting down the number of times a cell can divide before it enters senescence or apoptosis. This shortening is strongly linked to cellular aging.
That's the general picture in normal aging, yes.
But, and this is a huge but, in many types of stem cells, which need to divide continuously, and crucially, in the vast majority, maybe 85–90% of cancer cells, telomerase activity is found to be reactivated or maintained at a high level.
Absolutely. This reactivation of telomerase is considered a critical step in cancer development. It essentially grants cancer cells a form of replicative immortality, allowing them to bypass the normal limits on cell division and proliferate indefinitely, a key hallmark of cancer. This makes telomerase a very attractive target for potential anti-cancer therapies.
A profound biological trade-off, indeed.
What?
Immortality for cancer cells. It also really drives home the question. With all this replication happening constantly, and given that our DNA is under constant attack from the environment and even our own metabolism, how does it possibly protect itself from damage and errors? Errors that could lead to devastating diseases like cancer if left unchecked.
This brings us to the absolutely essential unsung heroes of our cells, the intricate network of DNA repair mechanisms. Our DNA is incredibly valuable, but it's also surprisingly vulnerable. It's constantly susceptible to damage. This damage can come from internal sources, like reactive oxygen species produced during normal metabolism, or errors during replication itself. Or it can come from external mutagens, things like chemicals in pollution or tobacco smoke, various forms of radiation, including UV light from the sun, and X-rays.
So threats from inside and out. What
Yeah.
kinds of damage are we talking about?
Oh, a whole variety. Damage can include chemical alterations to the bases themselves, like oxidized guanine, a common one, or the formation of cross-links between the two DNA strands, or bulky distortions like those thymine dimers caused by UV radiation, where two adjacent thymine bases get improperly linked together. And then there are physical breaks in the sugar phosphate backbone, either single-strand breaks or the much more dangerous double-strand breaks.
But yeah, lots of ways DNA can get damaged.
Hmm.
How do cells fight back?
Fortunately, cells have evolved an incredibly sophisticated and often redundant array of repair mechanisms to counteract these constant threats and maintain the integrity of the genome. We can broadly categorize them into a few key systems. First, there's nucleotide excision repair, usually abbreviated as NER. This mechanism is a specialist for fixing bulky lesions, distortions that significantly disrupt the shape of the DNA double helix.
Like those thymine dimers from sunlight.
Exactly. Thymine dimers are a classic target for NER. Another example would be large chemical adducts, where bulky chemical groups get attached to the DNA bases, like those from benzoaperine, a carcinogen found in tobacco smoke and burnt food. How NER works is quite complex, involving many proteins. Essentially, an enzyme complex recognizes the bulky distortion, then it makes cuts on both sides of the damaged segment on the affected strand, excising a patch of DNA usually around 20-30 nucleotides long.
Like it cuts out a whole patch.
Yes, containing the damage. Then a DNA polymerase comes in and fills the resulting gap, using the intact complementary strand as a pristine template to ensure accuracy. Finally, DNA ligase seals the remaining nick, restoring the original DNA sequence. And the clinical relevance here is stark. Defects in the genes encoding NER proteins cause the devastating genetic disorder xeroderma pigmentosum, or XP.
Ah, yes, XP. Extreme sensitivity to sunlight.
Extreme sensitivity. Individuals with XP cannot properly repair UV-induced DNA damage, like thymine dimers. As a result, they have a dramatically increased risk, hundreds or thousands of times higher, of developing skin cancer, often at a very young age. It tragically highlights the absolutely vital role of NER in protecting us from the damaging effects of sunlight.
A very clear and tragic link. Okay, NER handles bulky damage. What about smaller problems?
For smaller, more subtle lesions or abnormal bases that don't cause major distortions to the helix, we have base excision repair, or BER. BER typically targets things like damaged single bases, maybe an oxidized guanine, or a cytosine that has spontaneously lost its amino group, deaminated, and turned into uracil, which shouldn't be in DNA.
Uracil in DNA is a mistake.
Right. So BER starts with a specific enzyme called a DNA glycosylase. There are different glycosylases specialized for recognizing different types of damaged bases. The glycosylase cleverly cleaves only the N-glycosidic bond that connects the damaged base to the sugar, effectively popping the bad base off. This leaves behind an abasic site, specifically an apurinic or a pyrimidinic site, often just called an AP site. Basically, you have the sugar-phosphate backbone intact, but with a base missing at that position.
Just removes the base itself.
Exactly. Then another enzyme, an AP endonuclease, recognizes this AP site and cuts the sugar-phosphate backbone next to it. This allows other enzymes to come in, remove the sugar-phosphate remnant, have a DNA polymerase insert the correct nucleotide, and finally, DNA ligase seals the nick. It's a more targeted repair for specific base damages.
Okay, BER for small base problems. What about mistakes made during replication itself? The ones proofreading missed.
Ah, yes. For those errors, like incorrect base pairings, say a G paired with a T instead of a C, that slip past the polymerase's proofreading function, we rely on the mismatch repair system, or MMR. MMR's main job is to correct these replication errors after replication is completed but before the next round of cell division to prevent the error from becoming a permanent mutation. A really remarkable feature of this system, especially well-studied in bacteria but also present in eukaryotes, is its ability to distinguish between the original parental strand, which is assumed to be correct, and the newly synthesized strand, which contains the potential error.
How does it know which strand is which?
In bacteria like E. coli, it often relies on chemical markers, specifically methylation patterns. The parental DNA strand is typically methylated at certain adenine bases shortly after replication, while the newly synthesized strand remains unmethylated for a brief period. The MMR system recognizes this difference and preferentially targets the unmethylated new strand for repair. Eukaryotic MMR uses different signals, possibly involving nicks in the new strand, but the principle of identifying the new strand is the same.
Clever way to identify the mistake.
Very. Once the incorrect strand is identified, the mismatch repair system recruits enzymes to excise a segment of DNA containing the mismatch on the new strand, and then DNA polymerase fills the gap correctly, followed by ligation. And again, the clinical connection is critical. Inherited defects in human mismatch repair genes, like MSH2, MLH1, PMS2, are the primary cause of hereditary non-polyposis colorectal cancer. HNPCC, also known as Lynch syndrome.
Lynch syndrome, right. Increased risk of colorectal and other cancers.
Exactly. Individuals with Lynch syndrome have a dramatically increased risk of developing colorectal cancer, endometrial cancer, and several other types of cancer, often at an earlier age. This unequivocally demonstrates the crucial role of mismatch repair in maintaining genomic stability and preventing cancer development.
So NER, BER, MMR. These repair mechanisms truly are the unsung heroes, constantly vigilant, patrolling our DNA and maintaining the integrity of our genome against this barrage of daily assaults. It's quite incredible. But what about even larger scale changes? Things beyond single bases or small patches. What about major genetic rearrangements within or even between chromosomes?
Right. If we connect this to the bigger picture of genomic stability, genetic rearrangements represent more dramatic alterations, involving the significant exchange or repositioning of large chunks of genetic material. One major type is homologous recombination, HR. HR is a really critical process with dual roles. It's essential for generating genetic diversity during meiosis, the process that creates sperm and egg cells by allowing chromosomes to swap segments. But HR is also a major high-fidelity DNA repair pathway, particularly important for accurately repairing the most dangerous type of DNA damage. Double-strand breaks, where both strands of the helix are broken. HR uses an undamaged homologous chromosome or sister chromatid as a template to ensure the break is repaired correctly without losing genetic information.
So recombination can be good for repair and diversity.
Yes, when it works properly. But then there are other types of rearrangements that are often problematic, like translocations. Translocations occur when pieces of chromosomes break off and then reattach to a different location, sometimes to the same chromosome, but often to an entirely different non-homologous chromosome.
Swapping pieces between different chromosomes. That sounds risky.
It can be very risky. These events can have dire consequences, especially in the context of cancer. For instance, a translocation might inadvertently place a growth-promoting gene, a proto-oncogene, next to a very active promoter sequence, causing it to be constantly turned on, driving uncontrolled cell proliferation. The Philadelphia chromosome, seen in chronic myeloid leukemia, CML, is a classic example of this. Alternatively, a translocation might break right through the middle of a tumor suppressor gene, disrupting its function. Tumor suppressors normally act like brakes on cell growth, so losing their function can also contribute to cancer development. Such chromosomal translocations are unfortunately very frequently observed in cancer cells and are often key drivers of their malignancy.
Wow. So rearrangements can really mess things up. And then we have those truly intriguing, almost sci-fi sounding things, the jumping genes, transposons. These can literally move around our genome.
Yes, transposons, or transposable elements, are indeed mobile genetic elements. They are segments of DNA that have the remarkable ability to literally jump or move from one location to another within the genome. They were discovered by Barbara McClintock in corn, a finding that earned her a Nobel Prize.
Jumping genes. Amazing. Do they do anything useful?
They can be drivers of evolution by creating mutations and rearranging genomes over long timescales, but they can also cause problems. Sometimes they carry other genes with them. For example, transposons are notorious for rapidly spreading antibiotic resistance genes among bacterial populations, which poses a huge public health challenge. There are different mechanisms for how they move. Some transposons move directly as DNA, using an enzyme they often encode themselves, called transposis. Transposis essentially performs a cut and paste, or sometimes a copy and paste operation, inserting the transposon into a new genomic location.
Cut and paste DNA.
Others are retrotransposons, and these use a fascinating, more complex mechanism involving an RNA intermediate. First, the retrotransposon DNA sequence is transcribed into an RNA molecule. Then, a specialized enzyme, reverse transcriptase, often encoded by the retrotransposon itself, uses this RNA molecule as a template to synthesize a DNA copy. This DNA copy is then integrated into a new location in the genome, so it's a copy via RNA and paste mechanism. Lines and signs, like ALU elements, are common examples of retrotransposons in the human genome, making up a surprisingly large fraction of our DNA.
Reverse transcriptase. That sounds familiar. Isn't that what HIV uses?
Exactly. This mechanism is especially significant in understanding retroviruses like HIV. HIV is a classic retrovirus. It has an RNA genome. When it infects a cell, it brings its own reverse transcriptase enzyme, which converts the viral RNA genome into a DNA copy. This viral DNA copy is then integrated into the host cell's chromosome, becoming a permanent part of the host genome called a provirus. From there, it can be transcribed to make new viral RNAs and proteins, essentially hijacking the cell's machinery for its own replication. AZT, which we discussed earlier, works by inhibiting this crucial reverse transcriptase step.
Wow. So these mobile elements and viruses really highlight the incredible dynamic nature of our genome. It's not just a static blueprint. And it shows how these elements can be both drivers of long-term evolution and, unfortunately, agents of disease right now.
Absolutely. This entire discussion of replication and repair really underscores the incredible dynamism, the inherent vulnerability of our genetic material, but also the equally sophisticated and multi-layered cellular defenses that are constantly in place to protect our fundamental blueprint. It's a constant high-stakes biological arms race happening inside every cell.
Okay, we've explored how DNA copies itself with truly astonishing precision, thanks to polymerases and proofreading. And we've seen how our cells are constantly working nonstop, really, to repair any damage that inevitably occurs. Now, let's move to the very next crucial step in actually using that genetic information. How does the cell read the DNA blueprint to make something functional? This involves creating an RNA message from the DNA template, a process we call transcription. This takes us into chapter 14.
Transcription is, as you said, the synthesis of RNA from a DNA template. It's the absolutely essential first step in converting the static genetic information encoded in DNA, that A, T, C, G sequence, into a dynamic, usable form, typically an RNA molecule, that can then be used to direct the synthesis of a protein or perform other functions in the cell. This intricate process is carried out by large, complex enzymes called RNA polymerases.
RNA polymerases. Okay, similar to DNA polymerases.
Similar in that they synthesize a nucleic acid chain, but with key differences. Unlike DNA replication, which creates a double-stranded DNA copy of the entire genome, transcription typically only copies specific segments of DNA individual genes or groups of genes into single-stranded RNA molecules. And critically, only one of the two DNA strands, called the template strand, or sometimes the antisense strand, serves as the blueprint for RNA synthesis during transcription of a particular gene. The other strand, the coding or sense strand, has a sequence very similar to the RNA product, except for U replacing T. Also, another key difference, RNA polymerase can initiate synthesis of a new RNA chain without needing a primer, unlike DNA polymerase. And of course, as we established earlier, RNA polymerase incorporates uracil into the growing RNA molecule whenever it encounters an adenine A on the DNA template instead of thymine T.
No primer needed, uses U instead of T, and copies only one strand for specific genes.
Got it. What stands out to you about how this crucial process of transcription works in bacteria? How do these simple organisms manage to get their genetic messages transcribed efficiently? In bacteria, the system is remarkably streamlined and elegant, really built for efficiency. A single type of RNA polymerase enzyme is responsible for synthesizing all three major types of RNA: messenger RNA, mRNA, ribosomal RNA, rRNA, and transfer RNA, tRNA.
Just one polymerase does it all.
It's just one main type, yes. This bacterial RNA polymerase is a large multi-subunit enzyme. It has a core enzyme part, which contains the catalytic activity, the machinery for actually making the RNA chain. But crucially, it associates with another protein subunit called the sigma factor. The sigma factor is absolutely critical for initiating transcription correctly. Its job is to recognize and bind specifically to particular DNA sequences called promoters.
Promoters, like starting signals on the DNA.
Exactly. Promoters are specific DNA sequences, typically located just upstream, before the start, of the gene that needs to be transcribed. They act like molecular signposts telling the RNA polymerase precisely where to bind and where to start transcription. The sigma factor is what guides the RNA polymerase core enzyme to these promoter sites. Without the sigma factor, the core enzyme would just bind loosely and randomly to DNA and wouldn't be able to initiate transcription efficiently or accurately at the correct starting points. Different sigma factors can even recognize different sets of promoters, allowing bacteria to coordinate the expression of genes needed under different conditions.
So the sigma factor is like a highly specialized guide, making sure the RNA polymerase finds the correct starting line on the DNA track to transcribe the right genes at the right time.
That's a perfect analogy. It confers specificity and is a key point of regulation. And another feature often seen in bacteria is that functionally related genes are frequently grouped together on the chromosome into units called operons.
Operons, right, like the lac operon for lactose metabolism.
Exactly. The lac operon is the classic example. An operon consists of a set of genes, often involved in a single metabolic pathway or cellular function, along with the regulatory sequences, like the promoter and operator sites, that control their expression. These genes within an operon are transcribed together from that single promoter into one long polycistronic mRNA molecule.
Polycistronic, meaning one mRNA codes for multiple proteins.
Precisely. That single mRNA transcript contains the coding sequences for several different proteins. This setup allows the bacterium to efficiently turn on or off the entire set of genes needed for a specific task in a coordinated fashion. It's very economical. Transcription then proceeds along the DNA template until the RNA polymerase encounters specific DNA sequences that act as termination signals. These signals tell the polymerase to stop transcribing, release the newly made RNA molecule, and detach from the DNA. Termination in bacteria can happen in two main ways. Some termination signals are Rho-dependent, meaning they require the action of a protein factor called Rho, which somehow helps dislodge the RNA polymerase. Other termination signals are Rho-independent. These typically involve sequences in the DNA that, when transcribed into RNA, cause the RNA molecule itself to fold into a specific hairpin loop structure. This hairpin structure physically destabilizes the RNA polymerase's attachment to the DNA, causing it to fall off and terminate transcription.
Okay, Rho-dependent and Rho-independent termination. Clever ways to stop. That bacterial system sounds quite efficient, coordinating genes and operons. But how does that compare to us, eukaryotes? With our much larger genomes, our DNA packed away in a nucleus. What are the big differences in eukaryotic transcription? It must be more complex.
Oh, absolutely. If we connect this to the bigger picture, eukaryotic transcription is significantly more complex and involves many more layers of regulation. This complexity reflects our multicellular nature, the need for precise control of gene expression in different cell types and at different developmental stages. A major defining difference right off the bat is that eukaryotes employ not one, but three distinct types of RNA polymerases in the nucleus, each specialized for transcribing different classes of genes. RNA polymerase I. This polymerase is located in the nucleolus, a specific region within the nucleus, and it's solely dedicated to synthesizing the large precursor RNA molecule that gets processed into the major ribosomal RNAs, specifically the 18S, 5.8S, and 28S RNAs in humans. These are the most abundant RNAs in the cell, needed in huge quantities to build ribosomes.
Pol I for rRNA.
Caught it. RNA polymerase II, Pol2. This is arguably the star player, at least in terms of protein production. Pol2 is responsible for synthesizing all the messenger RNA, mRNA precursors, which in their initial unprocessed form we often call heterogeneous nuclear RNA, hnRNA. Pol2 also synthesizes most small nuclear RNAs, snRNAs involved in splicing, and microRNAs, mRNAs involved in gene regulation. So it handles all the protein coding genes. Pol2 for mRNA and some small RNA is a really important one for protein recipes. Exactly. And RNA polymerase III, Pol3. This polymerase synthesizes the transfer RNA, tRNA precursors, as well as the smaller 5S ribosomal RNA and various other small stable RNAs involved in different cellular processes. This clear division of labor among the three polymerases allows for much finer, more specialized, and independent control over the expression of different types of genes, which is crucial for eukaryotic complexity.
3 different polymerases for different jobs makes sense for more complexity. And you mentioned that Pol2 makes hnRNA. Unlike bacteria, this eukaryotic mRNA isn't just a raw transcript ready to go straight to the ribosome for translation, right? There's quite a fascinating amount of processing involved even before it can leave the nucleus.
Absolutely. This raises a really important question. What happens to that initial hnRNA transcript produced by RNA polymerase II? It's often much longer than the final mRNA and contains sequences that aren't part of the final protein code. This hnRNA undergoes extensive and absolutely critical post-transcriptional modification inside the nucleus before it can be considered a mature, functional mRNA molecule, ready for export to the cytoplasm where translation occurs. This processing is vital for several reasons. It increases the stability of the mRNA, it's required for its transport out of the nucleus, and it plays crucial roles in ensuring its efficient and accurate translation into protein by the ribosomes.
Okay, so what are these processing steps? You mentioned a cap and a tail earlier for mRNA.
Right. There are 3 major processing events that happen to most eukaryotic hnRNAs destined to become mRNAs. First, almost immediately after transcription starts, a distinctive 5' is added to the very first nucleotide at the 5' end of the growing hrNA molecule. This cap is chemically unusual. It's a modified guanosine nucleotide, specifically 7-methylguanosine, that's attached backward via a special 5' to 5' triphosphate linkage.
A backward cap.
Yeah, it's a unique chemical structure. This 5' has several vital functions. It acts like a molecular helmet, protecting the vulnerable 5' end of the mRNA from being degraded by cellular enzymes called exonucleases. And critically, it serves as the primary recognition signal for the ribosome to bind to the mRNA and initiate the process of translation in the cytoplasm. No cap, generally no translation.
Okay, capping protects the front end and helps ribosomes bind. What about the tail?
Second, at the other end, the thymidine, a poly-A tail is added. This happens after transcription is terminated. The hnRNA is cleaved at a specific site downstream from the coding sequence, and then an enzyme called poly-A polymerase adds a long stretch of adenine nucleotides, maybe 50 to 250 A's, onto this newly created 3' end. This poly-A tail isn't encoded in the DNA gene itself, it's added post-transcriptionally.
A long string of A's added on. What does that do?
Like the 5' the poly-A tail contributes significantly to the stability of the mRNA molecule, protecting its 3' from degradation. It also plays important roles in the export of the mRNA from the nucleus to the cytoplasm and in enhancing the efficiency of translation initiation. The length of the tail can even influence how long an mRNA molecule persists in the cytoplasm, acting as a kind of timer.
Cap for protection and binding, tail for stability and export. What's the third big step?
Third, and perhaps the most complex and fascinating processing event, is splicing. Most eukaryotic genes that encode proteins are not continuous stretches of coding sequence. Instead, they are interrupted by non-coding regions called introns, which are interspersed among the coding regions called exons.
Introns are intervening sequences, exons are expressed sequences.
Exactly. Introns can sometimes make up a very large fraction of the initial hnRNA transcript. These introns must be precisely removed, and the exons must be accurately joined together or spliced in the correct order to create a continuous functional coding sequence in the mature mRNA. If this isn't done perfectly, the resulting protein will likely be completely wrong or non-functional.
So the cell has to cut out all the non-coding filler introns and perfectly glue the important coding exon pieces back together. How on earth does it manage that with such precision?
With an incredibly sophisticated molecular machine called the spliceosome. The spliceosome is one of the most complex molecular machines in the cell. It's not a single enzyme, but a large dynamic complex made up of several small nuclear RNAs, snRNAs, remember, made by Pol II or III, that are associated with numerous proteins. These RNA protein complexes are called small nuclear ribonuclear proteins, or snRNPs, often pronounced SNRPs.
SNRPs, okay.
These snRNPs assemble onto the hnRNA transcript. They have the remarkable ability to recognize specific, short consensus sequences that mark the boundaries between introns and exons, the splice sites. Through a series of intricate RNA, RNA, and RNA protein interactions, the spliceosome brings the ends of the exons together, loops out the intron into a characteristic structure called a lariat, then precisely cuts the intron out and ligates or joins the two exons together. This happens for every intron in the transcript.
Forming a lariat loop. That's amazing. The precision required must be phenomenal.
It is. Even a single base error in recognizing a splice site or in the cutting and joining process can lead to aberrant splicing, potentially causing exons to be skipped or introns to be retained, or shifting the reading frame for translation. Such splicing errors are now known to be the cause of a significant number of human genetic diseases. The spliceosome's accuracy is truly paramount.
So the cell acts as this highly advanced molecular editor, meticulously cutting out all the non-coding introns and precisely gluing the important coding exons back together after adding a protective cap and tail before finally sending that refined message out to the cytoplasm to be read by the ribosome. There's just an astonishing level of quality control and engineering just to get a functional protein recipe. Do the other RNAs, like RNA and tRNA, also undergo similar kinds of processing in eukaryotes?
Yes, they absolutely do, although the specifics of their processing pathways differ quite a bit from mRNA splicing. For RNA synthesis by polyadenylation, it typically begins with the transcription of a single large precursor molecule, often called the 45S pre-RNA in humans. This large precursor contains the sequences for the 18S, 5.8S, and 28S RNAs, all linked together with spacer regions in between. This 45S pre-rRNA then undergoes a complex series of enzymatic cleavages and modifications, primarily within the nucleolus. These processing steps precisely cut out the mature RNA molecules and remove the spacer sequences. The mature RNAs then associate with dozens of different ribosomal proteins, which are synthesized in the cytoplasm and imported into the nucleus, to assemble into the functional 40S and 60S ribosomal subunits, which are then exported to the cytoplasm.
So RNA starts big and gets chopped up into the final pieces. What about tRNA?
Similarly, tRNA precursors synthesized by Pol III also undergo extensive processing to become mature, functional tRNA molecules. This typically includes enzymatic cleavage to remove extra nucleotides from both their 5' and 3' ends. Addition of a specific 3-nucleotide sequence, CCA, to the 3' end of all tRNAs. This is the site where the amino acid will eventually attach. And often, sophisticated chemical modification of some of the bases within the tRNA molecule. Dozens of different types of modified bases can be found in tRNAs, and these modifications are crucial for the tRNA's correct folding into its L-shaped 3D structure, for its stability, and for its accurate recognition by both the ribosome and the enzymes that attach the correct amino acid. Interestingly, some tRNA precursors in eukaryotes also contain introns, but these are removed by a completely different splicing mechanism that doesn't involve the spliceosome used for mRNA. It uses specific tRNA splicing endonucleases and ligases. It just highlights again how ubiquitous the need for RNA processing and editing is in eukaryotic gene expression.
Okay, widespread processing for all types of RNA. Let's try to bring this back to some practical, real-world scenarios again. We mentioned the antibiotic rifampin earlier in the context of bacteria. How does it tie into transcription, and why is it so effective?
Yes, rifampin is a really important antibiotic, especially for treating tuberculosis, which is caused by the bacterium Mycobacterium tuberculosis. Rifampin works by specifically binding to and inhibiting the bacterial RNA polymerase enzyme.
It stops bacterial transcription.
Exactly. By inhibiting bacterial RNA polymerase, it prevents the bacteria from synthesizing essential RNAs and proteins, ultimately killing them. The genius of rifampin, like many successful antibiotics, lies in its selectivity. It binds strongly to the bacterial RNA polymerase but does not significantly affect the activity of our own eukaryotic RNA polymerases, polymerase D2E2 at therapeutic concentrations. This fundamental structural and functional difference between the bacterial and eukaryotic enzymes is what allows rifampin to be used as a targeted drug, effectively killing the bacterial invaders without causing excessive harm to our own cells. It's a classic example of exploiting specific biochemical differences between pathogen and host for therapeutic benefit.
That's a great example of targeted intervention based on understanding these enzymes. But unfortunately, on the flip side, some toxins exist that specifically target eukaryotic transcription, and these can have truly devastating consequences for us.
Indeed. A classic and rather grim example is alpha-aminoitin. This is an extremely potent cyclic peptide toxin found in certain poisonous mushrooms, most notoriously the Amanita phalloides mushroom, which is commonly known as the death cap.
The death cap mushroom. Sounds ominous.
And it is. Alpha-aminoitin is deadly because it specifically and very tightly binds to and inhibits eukaryotic RNA polymerase II. Remember, polyadenylation is the crucial enzyme responsible for synthesizing all the mRNA precursors for protein-coding genes in our cells. By shutting down POL2, alpha-aminoitin effectively halts the production of nearly all new proteins. Because proteins are essential for virtually every cellular function, this leads to rapid and widespread cellular dysfunction and death, particularly affecting cells with high metabolic activity like those in the liver and kidneys. Ingestion of even a small amount of death cap mushroom can cause severe gastrointestinal distress initially, followed by devastating liver and kidney failure, often leading to death within days if not treated aggressively, example with a liver transplant. It tragically underscores the absolutely vital, irreplaceable role of RNA polymerase II in eukaryotic gene expression and survival.
Wow, a direct hit on POL2 is catastrophic. What's also truly fascinating, maybe in a less immediately lethal way, is how subtle mutations affecting these intricate transcription and RNA processing mechanisms can manifest as serious chronic diseases. For example, you mentioned thalassemia earlier. How does that relate?
Right. Thalassemia is actually a group of inherited blood disorders characterized by reduced or absent synthesis of one or more of the globin chains that make up hemoglobin, the protein responsible for carrying oxygen in our red blood cells. While some thalassemia mutations directly affect the globin protein sequence, many others are actually defects in gene expression. For instance, some mutations might occur in the promoter region of a globin gene, reducing the efficiency of transcription by RNA polymerase II, leading to less globin mRNA being produced.
Less message means less protein.
Exactly.
Right.
Or other thalassemia mutations can occur within the introns or at the crucial splice sites of the globin genes. These mutations can disrupt the normal splicing process carried out by the spliceosome. This might lead to introns being incorrectly retained in the mRNA, or exons being skipped, or the use of alternative incorrect splice sites. Any of these splicing errors will result in an abnormal, non-functional globin mRNA, which either gets degraded or translated into a useless or even harmful protein fragment. The end result, in either case reduced transcription or faulty splicing, is a deficiency in functional hemoglobin, leading to the characteristic anemia seen in thalassemia patients.
So problems in transcription or splicing directly cause the disease. That's a clear link.
Absolutely. And another really powerful clinical connection that highlights the importance of RNA processing comes from the autoimmune disease systemic lupus erythematosus, or SLE.
Lupus, right. That affects many parts of the body.
It does. SLE is a chronic autoimmune disease where the patient's immune system mistakenly attacks their own body's tissues and cells. Patients with SLE often develop a wide range of autoantibodies directed against their own cellular components. Crucially, many SLE patients develop autoantibodies specifically targeting components of the spliceosome. Remember those snRNPs, the small nuclear ribonuclear proteins? Antibodies against snRNPs, like anti-SM antibodies, are quite characteristic of lupus.
Their immune system attacks the splicing machine.
Precisely. By attacking the spliceosome machinery, these autoantibodies can interfere with the normal essential process of mRNA splicing throughout the body's cells. This widespread disruption of splicing can lead to the production of abnormal proteins, trigger cellular stress and death, and contribute significantly to the inflammation and diverse, often systemic symptoms experienced by SLE patients, things like joint pain, skin rashes, fatigue, kidney problems, and more. It's a clear illustration of how disrupting such a fundamental molecular process like splicing can manifest as a complex, multi-system disease.
Wow. Attacking the spliceosome itself. That really brings home how central these processes are. Okay, so we've covered a tremendous amount of ground today. We've journeyed from the structure of DNA and RNA through the intricacies of DNA replication and repair, and finally into the world of transcription and RNA processing. We've compared the elegant, maybe simplicity of prokaryotic gene expression with the profound complexity of our own eukaryotic systems. What does this all mean when we try to bring it all together? What's the fundamental takeaway, the big picture view, from contrasting prokaryotic and eukaryotic gene expression, tracing that path from DNA all the way to RNA?
That's the ultimate synthesis, isn't it? Trying to distill it down. I think the fundamental takeaways from contrasting prokaryotic and eukaryotic gene expression really revolve around increasing layers of complexity, compartmentalization, and regulation in eukaryotes, which allow for the development and maintenance of multicellular organisms with specialized cell types. Let's recap some key differences. First, genome structure and packaging. In eukaryotes, our DNA is linear, much larger, and intricately associated with histone proteins, forming structures called nucleosomes. These nucleosomes are further compacted into higher-order structures known as chromatin. This chromatin packaging provides a major layer of regulation. The chromatin must be dynamically unwound or modified, epigenetics, to make genes accessible for transcription. Prokaryotic DNA, in contrast, is typically circular, smaller, and lacks histones, though it has other organizing proteins, making it generally more readily accessible.
Chromatin adds complexity in eukaryotes.
Definitely. Second, gene organization. Eukaryotic genes are mostly monocistronic. This means one mRNA molecule typically encodes only one single protein. Prokaryotic genes, as we discussed, are often organized into operons, allowing a single polycistronic mRNA molecule to carry the genetic instructions for multiple different proteins that function together. This allows for very efficient, coordinated regulation in bacteria.
One message, one protein in eukaryotes, one message, many proteins in bacteria.
Generally speaking, yes. Third, compartmentalization. This is a huge difference. Eukaryotic transcription occurs exclusively inside the nucleus, physically separated from the site of translation, protein synthesis, which happens later on ribosomes out in the cytoplasm. This separation allows for extensive RNA processing to occur in the nucleus before the mRNA is exported. In prokaryotes, because there's no nucleus, transcription and translation are often coupled. They can occur almost simultaneously in the same cellular compartment, with ribosomes actually attaching to the mRNA and starting translation, even while the mRNA is still being transcribed from the DNA.
Coupled in bacteria, separate in space and time for us.
Exactly. And that separation enables the fourth key difference, RNA processing. As we've just discussed in detail, eukaryotic mRNA, from Pol to Dash, undergoes extensive post-transcriptional processing in the nucleus, the addition of the 5' cap, the addition of the poly tail, and the removal of introns via splicing. These sophisticated modifications are largely absent in prokaryotes, whose mRNA is generally ready for translation almost immediately after or even during its transcription.
That extensive processing is a hallmark of eukaryotes.
It absolutely is, and it provides many additional points for gene regulation and also allows for things like alternative splicing, where one gene can actually produce multiple different protein variants by splicing the exons together in different ways. That greatly expands the coding potential of our genome.
It's truly a masterclass in biological engineering, isn't it? From the elegant, fundamental structure of the DNA double helix, all the way through replication, repair, transcription, and processing. It's an intricate symphony of enzymes and processes that bring genetic information to life, meticulously building everything from a single bacterium to a complex human being. The sheer ingenuity, the elegance of these molecular mechanisms is just awe-inspiring when you really think about it.
Absolutely. We've traced the flow of information today. We've seen how the very chemical structure of DNA and RNA dictates their unique functions and interactions. We've explored how DNA is meticulously replicated with incredible fidelity thanks to polymerases and proofreading, and how it's rigorously repaired when damaged, thanks to sophisticated surveillance systems. And we've delved into how specific genes are selectively transcribed into RNA messages, which are then carefully processed and edited. Each step in this central dogma of molecular biology is truly a marvel of biochemical engineering, and as we've seen, it has profound and direct implications for human health and disease.
Yeah, and the clinical connections we've highlighted throughout this deep dive, they really drive that point home. From intelligently designed drugs like AZT that target viral replication by mimicking DNA building blocks, to antibiotics like rifampin that specifically shut down bacterial RNA polymerase, to devastating inherited genetic diseases like xeroderma pigmentosum caused by failures in DNA repair, or thalassemia resulting from defects in transcription or splicing, and even complex autoimmune conditions like lupus that can arise when the body mistakenly attacks its own splicing machinery. All of these examples powerfully underscore just how vital these fundamental biochemical processes are to every aspect of medicine.
Understanding these mechanisms at this level of detail not only illuminates the intricate origins of disease, why things go wrong, but also, critically, it provides the precise molecular pathways and targets for developing rational therapeutic interventions. The delicate, dynamic balance of DNA replication, DNA repair, transcription, and RNA processing is absolutely crucial for maintaining life. And when this balance is disrupted, whether by mutation, infection, or environmental damage, it often points the way towards diagnosis, prevention strategies, and targeted treatments.
So what does this all mean for you, listening right now? It means that every single second in trillions of cells throughout your body, every time one of those cells divides, every time it needs to make a specific protein to carry out a function, there's this astonishingly intricate, highly regulated molecular dance happening. A dance that keeps you alive, allows you to grow, to think, to function. And when something goes awry in that delicate choreography, a mutation slips past repair, a virus hijacks the machinery, transcription gets dysregulated. The detailed understanding of these very processes we've talked about today is precisely what allows scientists and clinicians to figure out what's wrong, to potentially intervene, to develop new therapies, and ultimately to advance medicine and improve human health. Just think about it. Nearly every drug you might encounter, almost every disease state, fundamentally connects back to the integrity and proper functioning of these pathways governing our genetic information. It's an incredibly robust system, built over billions of years of evolution, yet as we've seen, it's also beautifully vulnerable at specific points. Understanding both its strengths and weaknesses is really the key. What an amazing area to study. Thank you so much for joining us on this deep dive into the core of gene expression and protein synthesis. Keep that curiosity alive, keep asking questions, and keep exploring the incredible wonders within your own cells. We'll catch you on the next one.